Okay. Let me check for you. Uh, yeah, I think we are both live on YouTube as well as in Teams. Good evening, everyone. Welcome to my first live session in this session of data analysis for biologists, January 2024 session. I welcome you all to this live session. You must have gone through two other live sessions conducted by our PMRMTA on live problem solving session. I hope you have joined it. I am Biplab. I am the course instructor for this. Me, along with my TA here in IIT Guwahati, Oro, and Urpit, the PMR TA, are running this course this time. I hope you are already 3 weeks has passed. I hope you are enjoying the course and you must be learning new things, recapitulating the old one, and I'm sure you are quite excited for this one. And hopefully you have also registered for the final certification examination. So what do I do in this particular 1 hour today is that I will discuss some essential aspect, logistical aspect that you should keep in mind while going through this course, right? How to prepare for exam, how to do the assignments, all these questions. What you should keep in mind during the assignments and exams. Second thing is I'll also highlight certain points that when you are studying this course for this based upon this last 3 weeks experience, the points that you should focus on, right? And then if you have any queries regarding in general about this course, scope of this course, as well as any particular topic that you are finding difficult to understand, or you want us to explain that in details, I'll try to explain it here. If it is a lengthy one which requires much explanation, then I can post it on the forum also, or I can ask our PMRFTA who will be taking in this Saturday also one live session to explain that in detail, because that is usually a 2-hour session, right? So let me start with this one important point. Please interrupt me anytime. You can raise your hand. We are in Teams. If you are joined to Teams, you raise your hand. I will allow you to speak. You can ask a question. Or if you have joined through the YouTube, please use the chat option. Post your questions there, right? Anytime you interrupt, you have not to wait for me to stop and then ask, right? I will try to answer whenever possible in between. So, you must have a broad idea that what this course is all about and why you want to join. One important aspect that we have to remember that any science, whether it is physics, chemistry, biology, it doesn't matter, anthropology, any economics also people will make, some people will consider science, any field of science depend upon observation, empirical observations. You collect data, you do experiments, you collect data from the field, epidemiological data. Or suppose you are you're a physicist working on astronomy, you are collecting data, astronomical data from your telescope. So data is the key of any sort of science. Now, once you have the data, you are into multiple issues crops. One is any sort of data that you handle has some sort of noise, some spurious information in that. That will always have some sort of bias. One first step comes is to get rid of those noises, those biases from your data. Then come the analysis to check certain hypotheses. When you want to analyze them, then you want to also visualize the data. You want to look into different aspects of the data. So two, three components come here. One is statistical analysis. The second one is data transformation, like creating models like regression model, classifier, something like that, and data visualization. So if you look into biology curricula across the globe, statistics is usually one of the component at different level. One student, in some university curricula, statistics may not be in so depth, but at least superficially, we try to teach people of every field of science, including biology, some sorts of statistics. And if you are from the biology background, then we call it biostatistics. But remember, this course, data analysis for biologists, is not a statistics course. Statistics course, biostatistics, if you look into the core concepts of biostatistics, what you are doing there, you are trying to calculate statistics of your data, mean some sort of compilation of your data. You calculate mean, you calculate variance, you calculate median, we call them statistics. So you are, extracting the essential features from your data. At the same time, one very important aspect of statistics is of hypothesis testing. To ask question, what I am seeing, is it correct or it is just a spurious? This type of question you ask, right? You have treated animals with a drug and you have seen the animal is recovering. So you ask the question, is it just a placebo effect, just by chance it has happened, or it is really the drug is working? So you do test of hypothesis. These are classically part of statistics. In this course, we are not going into. What we are discussing now are essentially part of what in large context called machine learning. You want to create some sort of model, some sort of classifier to understand the data, extract a meaning out of that. Now remember, almost all fields of science and engineering now use this sort of machine learning algorithm, whether you are creating a linear, non-linear model through regression, or you are creating a classifier using a tree-based method or something else. It doesn't really matter. All this comes under. Can you hear me? Can somebody confirm? Voice is me getting muted and I'm getting removed of the meeting. I think there is an issue of rights you have given to people. I was removed from the meeting, so I'm joining again actually. Give me a minute, I have to share my screen. So if anyone there in our guest list who has somehow got the option to unmute me, can mute me, please don't do that mischief, because that doesn't help anybody. So as I am talking of, I hope you can hear me now. So what I was talking is that so this course primarily focused around this machine learning algorithm. We will deal with regression model, linear, non-linear, polynomial regression, different regression model, where you want to create a model out of data. Then we will look into the problem of classification and clustering. You want to get cluster out of data. You want to do that. You want to find classified data. You have gene expression data, suppose microarray data or single cell RNA-seq data. Based upon that, you want to classify people, patients, whether they have the metastatic cancer or benign tumors. So you require classification models, right? So we call them classifier. So we'll go through one or two different types of classifier. Remember, these are also machine learning algorithms. At the same time, we'll also go through one particular important thing called data dimension reduction. Because nowadays, biology, whether it is coming from epidemiological data or your molecular biology data, we are using high throughput experiments in molecular biology. So you are generating data which is a very high dimension. And I will discuss in this course, what do I mean by dimension? Like, it is not the issue of number of samples, it's the issue of variable. You have a large number of variables and classical statistical method fails. So we have to reduce the dimension. That's a very important thing. And once you reduce the dimension, you have to visualize them. Along with it comes obviously data visualization. So in these 3 weeks, we have not delved into those things. The core machine learning thing we have not delved into. I'll remind you the techniques and methods that we discuss in this course are not unique for biology. These are generalized methods used across different fields of science and technology. The only thing is what I have done in this course is that I have picked up examples to explain them based on biology example, as simple as that. If you are coming from a different field or you want to use it in different field, you can happily use it if you understand the method or the algorithm. You can easily use it in some other field also. Now, in the last 3 weeks, you must have seen we have not discussed any of this. What we have discussed is in the first week, I have discussed about basic concept of, what we call, let me take my pen. We have discussed about basic concept of probability and statistics. And then in the next week, we have discussed linear algebra. And in third week, we have started R. Now, you must have understood why R is required. See, R started as an open source movement part for data analysis, large scale data analysis, machine learning, model building, right? These languages was developed as an open source thing, but eventually right now, this is an industry standard. And we have a huge library, and every day the library is increasing, which helps us to analyze different sort of data using different sort of the state of the art algorithms. So if you are in data analysis, you cannot escape R. You have to learn how to use R. So in this course, we insist that you learn R step by step. In week 3, we are introducing you to the basic things of R. I understand many of you may not have done any programming earlier, but believe me, without using R, using Excel or Sigma plot or something like those tools, you cannot do large-scale data analysis that we will teach in this course. So if you are a researcher or you are planning to go into industry, you want a data analysis position, or you want to increase your portfolio by saying, improve your portfolio, saying that you know some aspect of data analysis, you need to know R, right? So I hope you people have installed R in your machine and started learning. Now, some of you must be wondering that why the hell I have to learn probability, statistics, and linear algebra. The fact is, we are in 2 weeks. We have in 1 week, we have some lectures on probability theory, and in 1 week we have linear algebra. These are just scratching on the surface. The main issue is every algorithm, every method of machine learning, starting from regression-based model to clustering, classification, dimension reduction, whatever you learn, everything relies on concepts of probability and linear algebra. Now, if you do not understand those basic terminology, then you are in trouble. I can teach you how to press switches or copy paste some R code, but that will not help you. That may help you to, analyze one set of data. That may help you to create some pretty pictures, graph maybe, but that will not help you in long run because you will not understand 2 things. One, what is the meaning of that data? And when not to use a particular algorithm. These 2 are major problems that we see among students and even in citizen scientists. If they do not understand, if a student or a scientist does not understand the basic physical meaning of the algorithm, then they misuse it. One common problem in biology is, for example, misuse of t-test. T-test can be done very easily. In Excel also you can do that. And so every time people get data, they try to do a t-test, even when t-test is not the right choice. Why they fail to understand? Because they have not got into the physical meaning of the t-test. What is meant by all these mathematics? That is what the problem is. Very recently, I observed, I was reading a paper where they have used dimension reduction technique called PCA, which is completely based upon probability theory and linear algebra, and they have completely wrong explanation of the PCA result. So all these things crops up because the person who is using these methods, which are very powerful, and you have ready-made script to use and analyze using those algorithms, are misusing them because they don't understand the basic concept. So we are teaching you the essential part of probability theory and linear algebra that will help you to understand the algorithms of regression, dimension reduction, clustering, classification, the meaning of those things. right? Not in detail. I am not expecting that somebody of you will become a mathematician out of this course. That is not the purpose. The purpose is you understand what do I mean by regression. When regression can be done, what is the meaning of statistical test of the regression coefficient? Why that those are required? What is the meaning of the new dimensional coordinated PCA? Those meaning, if you have to understand, you require probability theory and linear algebra. And if you understand the physical meaning of those, you will never do a wrong interpretation or misuse of them. And then we'll go into data visualization in the 4th week. 5th week, we'll start regression-based model, linear, non-linear, all these things. Then we will enter in the 6th week, the classification and clustering algorithm. Fine. It will continue in the 7th week also because multiple algorithms are there. And in the final week, we'll deal with higher dimensional data, like coming from single cell RNA-seq data or microarray data, how to reduce the dimension. You may have seen pretty picture of Disney or diffusion map or UMAP, all these things. We'll learn those algorithms. Okay, if anyone has any question up to this, you can raise your hand here or post in the chat in the YouTube. Okay, let us go into some logistical issue. I do not see any question. If you have, you can raise your hand in Teams or post on the YouTube chat. So obviously, we have multiple type of live session. For example, this one I am doing, and maybe we will have another live session with me, depending upon the time slots available. But one thing is for sure, every week Saturday, we have the PMRFTA problem solving session, where Harpreet is dedicatedly solving through all the questions asked in the the lecture videos, as well as I am providing you additional questions which is solving step by step so that you can understand how to solve the question. Also, he is trying to make a précis of each lecture. And in this session is a 2-hour session, so you can clear all your doubts. So I will insist, please join. Yes, these online sessions, these live problem solving sessions are recorded and provided in YouTube. That means you can watch them later on. But remember, if you are not present in the session, you cannot ask questions. So the best way, see, we all understand, you have to remember, we all understand that online learning is not easy. Online learning, in normal learning, you have a class, you go there, somehow even in 1-hour class, even if you pay attention for 10 minutes, you learn something there and then. If you have any query, you can immediately ask the instructor. In online learning, you don't have these options, right? These are pre-recorded videos and the lecture slide. You are watching them, going through them in your extra time that you can spare. Now, obviously, you will get lots of questions. Many ideas does not get clear to you. The best option to get those cleared is the live problem solving session. Join them, ask questions. So what you do, it is every Saturday at 6 o'clock. The schedule is given there. The Teams link is also there in the announcement section. You go to the announcement, you get the link. Before that day, why don't you go through all the lectures of that week and jot down your questions, queries, difficult part that you have faced, and ask those questions during the live session. I can guarantee then you will go through this course, sail through this course very easy, and also learn the subject, right? So I insist that please join. Do not rely on this that okay, anyway, this will be posted on the YouTube, so I can watch it later. Yes, it will be posted on YouTube. You can watch it later, but that does not help actually if you are not present in the live session. So what I see, we have some question here. Before I move, let me ask: will this Simoy is asking? Will this live be saved on the channel page for future reference? Yes, these are saved and posted in the YouTube, so you can go through it. Surya has asked, how R is different from Python? I haven't attended these week lectures, so maybe it will be repeated question. Okay, means I do not understand really what do you mean by what is the difference, right? Obviously, Python and R are two different languages. Both have been created ground up with a completely different purpose. Python was not really originated for this type of scientific data analysis. It originated from some other purpose. Although now Python has completely diversified, and if you are working on AI, Python is your bread and butter, right? But you have to remember, R has been created bottom up, right? At the machine level, when you develop a language and you work on the machine level, so it has been developed bottom up for data analysis, particularly scientific data. So that's why the majority of the scientific data analysis algorithms are obviously in R, and are do a fantastic job in terms of scalability of the data analysis. Means how big data set I can analyze easily, how it can handle, what is the performance, those issues are there. So that's why R is a gold standard in large scale data analysis. Right now, in other form of how R is different, obviously how you index is different. Indexing in R is different in Python. I mean, I am talking of the syntax. So syntax between R and Python are slightly different. Several differences are there. But if you know one programming language, it is not so easy to migrate to the other programming language, right? That's what we all do. Most of us learn multiple programming language. None of us are master of all languages. We use them depending upon our purpose. So if you have already done something like you have done C, Fortran, even Fortran, C, or you have done Python, learning R will not be very difficult for you. There is a question here in the chat. Let me take that. Shika will use R in this course. So third week lectures are all for warming up with R. Fourth week lectures are all data visualization using R. So I will insist, please install R in your computer, desktop or laptop, whatever you have, and start learning. Remember, we are providing all the codes there, that the codes that I use in my lecture are all given through the portal, as well as I will share the GitHub link. I have posted the GitHub link in the discussion forum. You can download the codes, play with them. And remember, learning a language comes from playing with the language. You take a code, edit it, change it, think about it. Suppose somewhere there is a colon. What happens if I remove the colon? You do it, you see what happens, what error comes in the screen. That is the best way to learn. Fine. And there is another question. Sir, I am a biochem undergrad and I want to know what should I study to pursue masters in public health and epidemiology. I have no knowledge on this topic. So I am the absolutely wrong person to answer this question. I'm sorry. You have to ask somebody who works in the field of public health. Okay, so so far we have discussed our live session. Now let me check, is there any other questions here? No. Okay. Okay, so let me now go to another important topic, which is all about R. So what are those? As I said, learning R is essential, right? Now, many a times people ask this question, will there be any R in the final examination? No. The final examination is like your GATE and JEE type, right? Online examination, where you have this software on your computer where you are sitting in the exam hall. You will not get R there. So there will be no question where you have to program using R. No. But there will be R-based question, right? What do I mean by R-based question? So for example, suppose we will say, okay, I am giving a R script. A is equal to AZ2 and B is 4 star A. Then this script is given and then the question is asked. In a question paper, this script is given and a question is asked, what is the value of B? To answer this, you have to understand how R script works, right? So this type of question will be there. And this type of question will be there from all the topics that we are discussing, because third week onward, we are continuously up to 8th week will continuously use R, right? So go through those are script that I am using in my lecture, and I already shared them. So you go through them and learn how to use them, right? So you have to learn R. Without that, you cannot help. Okay, as I said, so you can download it from the course portal. If you log into the course portal, suppose you are in a particular lecture, you have the YouTube video here, right? And then you have some text below it explaining what is there in the YouTube video, and there you will find a download link, right? So that link in some lecture is for the slides, but in some cases that also include the code that I have used in that particular lecture, right? So please download. If you want to download all the codes used in the lecture at one single go and save it in your computer and use it, practice it, play with it, you can go to my GitHub page, right? And you can download it easily, right? This link is here, you can copy it. Otherwise, this link I have posted in the forum. You can use that link from there also, right? So you please download it and use it. As I said, the best way to learn a programming language is to play with the ready-made and see what is happening. Otherwise, if you believe that you will take a book and step by step everything you will learn, you cannot do that in this short period of time, right? So now another question comes very frequently is the calculator. If you have done GATE or JEE type exam, you know you cannot use a calculator. You cannot carry a calculator in the exam hall. The software of the examination thing, you will have a scientific calculator and you have to use it. Second thing, in many a time, for example, in the week 2, where we are calculating eigenvectors. So I have asked sometime that in the lecture also to use online tool to calculate eigenvectors, right? But remember, these type of online tools will not be available to you in your lecture, exams. Now that means I will only ask questions which can be manually solved by you. Remember, that means I will ask you question in exam, the final exam, which can be manually solved by you by paper and pen using the scientific calculator provided in the computer, the exam software, right? So that means you should practice some of them. Now, where obviously I will not ask you to use a method which I have not taught in the lectures. So suppose you have to calculate determinant. Now, if the matrix is very large, manually you cannot calculate the determinant. You may require an online calculator. So obviously nobody will ask in an exam a large matrix determinant. But determinant of a small matrix, 2 by 2 matrix, can be easily solved manually. Inversion of a 2 by 2 matrix can be easily done manually by paper and pen. You don't need a scientific calculator also. So practice those. So whenever you are going to this lecture, think about what are mathematical problem can come from these lectures, which I can solve by paper and pen. Try to do them paper and by pen. Also look into this assignment. Go through the extra question that we are discussing in the live session. You will see most of them can be solved by paper and pen. Try to solve them paper, right? One question is there. I am doing a metagenomic project from my thesis. I am planning to implement random forest model, but I am in scratch. Please go starting point. How can I learn in a proper way? So we will have it in the 7th week. I think we will have the random forest algorithm. So you have to wait up to that. So I will insist, please wait up to that. Go through step by step, because to understand random forest, you need understand decision tree, and also learn some sort of, what we call these basic probability, linear algebra, all these concepts. So go through this step by step. In the week 7, we'll learn decision tree classifier and the random forest. I hope you will be able to pick up from there. Although you may not be using exactly the metagenomic data or something, but the key concept, the key ideas will be made clear to you. How they work, it will be clear to you, and then you can use a ready-made algorithm in R and try on your data, right? But remember one important thing, as I said, understanding what is random forest, what is its limitation, when it can be used, what is the meaning of the data is more important, right? Otherwise, anybody who can learn English, I can teach to press some key and write some text and copy paste some text and do the data analysis. For that, you do not need a graduate student or postgraduate student, right? So the expectation is you learn. So wait till up to week 7, it will be clear. What kind of question can we expect in final examination? Will they be similar to assignment and practice question? Obvious. The questions will be similar to on this assignment and the practice questions. All will be either multiple choice or short answer. Short answer means the numerical value you have to write, right? So this is a standard way that online exam happens. In other online exams happens also. So you can easily expect that yes, some of the questions will be easy, some of the questions will be in between, some of them will be bit difficult, so that we can have gradation. But I think over the year I have seen students perform very well in it. The only requirement is you are consistent. Means you go through the course, you submit all assignment. That is one important thing. I have many times I have seen people do not submit all assignment. Don't do that. A large chunk of marks come from the assignments, right? Submit the assignment in time, right? Assignments are the best way to get the marks. So you submit the assignment, go through the problem discussed in the live sessions, right? And I'm sure you can sail through. The questions will be similar. Hope this is clear. Okay. So now let me go to some other issues. If you have any query, please post it here. Okay. So week 1. Okay. Week 1, basic concept probability testing is so hard. What type of question will be there in exam? Okay. Now I understand for some people it may be hard, but as I said, we need to understand the the basic essential understanding of probability, right? So how should you get? How should you get? If you are finding it hard, you are not alone that who is finding it hard. Many people may be finding it hard. For some people who have left mathematics long back, it is hard sometimes because the symbols become a bit difficult to understand, right? My suggestions are very clear. Please go through each video multiple times. Download the slides, go through the slides. This is your part that you have to do. Go through them. Note down which part you are not understanding. Then you have multiple options. Post the question on the forum, right? Post the question on the forum. Some of you, there is nothing to be shy actually. Means if you believe everybody who has joined this course know everything, no, not really, right? You may not know, maybe a large number of students are like you in the same problem, right? So if you post the question there is no silly question. Every question is important for us. So post the question there, and we usually answer within 1 day or maybe on the same day we try to answer. The second thing is, if you are really shy, you are, you don't want to let others know that you are failing to understand this particular thing, no problem. We are always available to you. My email ID is in public domain. Please send it to me, email. Either I will answer or the course TA or Oro will answer you. Be assured, we will take every step so that we can make you understand. But the only thing you have to tell us that I do not understand, right? You have to approach us. Otherwise, I will consider that you already understand. The third option you have, ask question during live session, including this session also, right? Now, what type of question you can ask? Explain. The questions would not be, based upon what is in the lecture videos. No question will come beyond the lecture videos. You don't need to read any book, any additional material for this. Every question will come from the question videos and the the R code that we use. So if you go through repeatedly, you can easily answer everything. Okay. Particularly what type of question? There will be MCQ and small short answer type in short numerical value type. Just now you have to calculate the value and write it, right? So look into the assignment questions. Look into the questions that we are giving in the additional question in the live session. Note down these questions. So if you can answer those type of question, and if you have gone through each and every line of the lecture videos, you can answer in every week course, every week of this course, as well as in the probability, right? So I cannot see your full name who has said that you are finding it hard, but don't feel shy. If you had to post 10 questions in a week to me, post 10 questions. We'll be happy to answer your question and clarify the doubts. Fine. You can ask the question here also. There's nothing to be ashamed of asking question, or you can post on the forum. I hope this helps. If you are not forthcoming, I cannot help you. That's why I'm explaining. Okay. So continuation to this, if I say in the first week, what you should learn? Basic rule of probability. Basic rule means okay, addition, multiplication. When you add, when you multiply probabilities, these are very essential things, right? This is not essential only for data analysis, for any scientist. You have to know this, right? Then there is the idea of mutually exclusive and independent. We have explained in this video. Remember that these things you have to understand. What is independent? What is mutually exclusive? And when it is independent, how the probabilities are calculated. In mutually exclusive, what happens to the probability? So all these things are in the lecture. Then the question comes of PMF and PTF. These are connected to two important concepts: discrete variable, discrete random variable, and continuous random. Many people wonder why random variable we are talking about. Actually, anything that we measure, mathematically we consider them as a random. Anything you measure. You are measuring my blood pressure, we will consider them as a random. The blood pressure can be any value, right? From it can be 120, it can be 121, it can be 121.5 also, right? So it's a continuous random value. Whereas if you are measuring something which will be discrete values like 2, 3, 4, 5, it cannot be 2.2. So that is a discrete random value. So anything you measure in experiment are random variable, and they are of two types: discrete and continuous. If you are dealing with discrete random variable, that is your discrete variables you're dealing with, then the concept of PMA will come to calculate the probability. Whereas if you are dealing with, continuous random variable, the PTM. So there are some standard distributions which are very useful: binomial, multinomial, Poisson. These are used every day in biology. Every day in biology, you may be also using them without knowing actually, right? So you learn about the PMF with this. These are for the discrete one, right? Whereas normal distribution, standard normal distribution, believe me, if you are using any statistics in your work, you are already using them. Unfortunately, possibly without understanding the meaning of it. And then we learn about uniform distribution, exponential distribution, and their P. So in both these cases, we learn about the mean and variance of those. So for example, we know how to calculate the variance of a Poisson distribution. In that case, variance and mean is same. It is given in a table in the lecture. So you can expect a question. If the mean of this Poisson distribution is 20, what is the variance? Answer will be 20. So that's what I'm saying. Everything is there in the slides and the lecture. All questions comes from there only, nowhere else. Okay, then we learn about mean, variance, moments, right? And then how to calculate them with Bessel's correction from the data. We learn about covariance. Understanding of covariance is very important, because from this covariance later on we will go into correlation, which will be used to create model, linear model. You must have done linear regression when you have taken, standard curve. For example, suppose you are doing protein assay. Using UV visible spectroscopy. Colored solution are obtained by adding some reagents to the protein solution, and then you use a standard curve. So for the standard curve, you have known amount of proteins and known concentration of protein samples. You do the same experiment, you get different color, calculate the absorbance, and you make a plot. And you get data point, you feed them in a straight line, saying that these data follow Beer-Lambert law. You call it linear regression. You may have done it using Excel sheet also. Now, in that case, the correlation, coefficient of determination, all these things are important. Again, those are all based upon covariance. So step by step we learn. We learn covariance first. We understand what the meaning of covariance. What does it tell me about linearity in the data? Then when we do linear regression, we will come and say, okay, what is the problem in covariance? Okay, there is a problem, so I require correlation. Then from correlation, then we go to coefficient of determination. So all these things are connected. If you miss one, you miss everything, right? Okay, then one part where we learn about contingency table to calculate marginal, joint, and conditional probability. Practice this. These are across all competitive exams, even related to biology 1. This is where table you get very frequently you get questions. A table is given, calculate the calculate some probability. Very easy to do if you understand once. As I said, if you do not understand, go through it once again, go through our lecture video. Still, if you do not understand, pose the question. Then Bayes theorem. Bayes theorem is essential of any biologist. Think about the dispute when the COVID test was developed during the corona, the COVID thing. So all diagnostic tests have issue of specificity, sensitivity. Some of you may be working on the field of biosensor. Some of you may be developing new assays for some disease. All these assays, all these sensors, you have to calculate specificity and sensitivity. And the specificity and sensitivity, from that you have to calculate what is the predictive value of your assay. Without that, nobody is going to, believe in your work. You require the Bayes theorem. Okay, there are calculators by which you can calculate it, but if you understand it, then it becomes more meaningful. So understanding is not so easy. Bayes theorem is very easy. We have discussed that. You can understand specificity, sensitivity. You must learn. We will come back to them again when we will do classifier. Because classifier, just like diagnostic test, just like a ELISA test, classifier also have sensitivity, specificity. Same thing will be used. Same Bayes theorem will be used. So you have to understand what is sensitivity, specificity, what is the different name of sensitivity, specificity. All these things are given in the lecture. Don't miss them. Then the last one, although is not required in extensively in our course, that's why we have discussed only t-test. You understand the basic concept of null hypothesis and alternate. Because we will not go into detail biostatistics. As I said, this course is not a biostatistics course, but you need to understand null hypothesis and what do we mean by test of hypothesis. Because suppose you are doing a simple linear regression, the same thing, concentration versus absorbance. Once you have done the linear regression, you are supposed to use some statistical test. I know many of you may have done the regression and never done the statistical test, but that's wrong, actually. We are supposed to do statistical test after linear regression, non-linear regression, every time. So to understand why we are doing the statistical test, we have to understand what is null hypothesis, what is meant by the statistical test of hypothesis. That's why the last lecture is there. There are ready-made tools to do that in R, but you have to understand the physical. Okay, there is a question you have asked, what type of question, objective question will be, you have objective question. Okay, yes, objective question. The type of question that we are asking in our, what we say, assignments, you will have the same type of questions. It will be MCQs or short answer type. There will be a place where you have to write the numerical type, right? So suppose I ask you to calculate a particular thing using suppose this specificity sensitivity issue. A numerical value will be suppose 1. So you have to write 1 something, right? So these two type of questions. So if you are doing assignment, you can easily understand what type of question. Okay, so the second week, vector matrices. I hope this is not difficult for most of you. It's very easy. You have to learn what is vector matrices because when you work in R, everything is either vector or matrix, actually, right? So you have to understand how to manipulate those data, vector and matrix. A whole table, you may be saving your data in a table in an Excel file, right? But when I will work in R, I'll convert that into a data frame, and that data frame is equivalent to a matrix. Each column is a vector, or each row is a vector. And then when you rearrange data, transform data, what you do is nothing but vector and matrix operations. So that's why this idea of addition, multiplication, dot product, projection, unit vector, orthogonal, these are important. So please go through these ideas. Then different types of matrices: square, rectangular, identity, diagonal. These are very important for data transformation and other algorithms. As I said, almost all algorithm in machine learning is based upon linear algebra. Then some simple matrix operation: addition, multiplication, transpose, determinant, inverse. As I have written them in red, practice whatever matrix operation we have taught you in this course, at least for 2x2 system using paper and pen, right? And also practice using R. So you take a small vector. For example, you do a matrix. Suppose you take 2, 3, 4, 5. You want to calculate the determinant of that. You can manually do that. You can do a, you want to invert this matrix. You want A, so you want to calculate determinant of it. Practice it by manual. You learn how to calculate the inverse of this matrix. It's 2x2 matrix. I have shown it in the lecture how to do it. You can do. Then create an arbitrarily large matrix. Suppose for example 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Now you cannot do it by paper and pen, right? So use R. I have shown that how to do that in a third week also. So in this way, practice both of them, right? Then we'll go through linear system of equation. Without these, actually, you cannot understand linear algebra, linear regression, non-linear regression. So you have to understand how a system of equation is converted into matrix format and when I can solve it, when I cannot solve, right? Because over-determined system, I have discussed in the lecture, all regression problems are over-determined. So you cannot solve them, but you cannot do regression. So to understand the difference between solution of a system and regression model, you have to understand this. And then this idea of linear independence. Lots of people has confusion on this. Good thing is that they are posting the question in the discussion forum. And we have answered them with example. We have explained what is the meaning of linear independence, how does that affect rank and all these things. If you also have confusion, please go through them. All those answers in the forum, all these questions will be made. The last one in week 2 is SVD. It may be difficult for many of you, but don't go mad on that. Just superficially also you can go through it. We understand SVD may be difficult for you. I have added it in the course because SVD is a technique which a large number of machine learning algorithms that we use are actually using in the back end. Although you don't use it directly, in the algorithm is actually doing. So just to as a flavor, if you tomorrow appear for an exam, interview where some data analysis question comes and somebody try to pull your leg saying, do you know SVD? At least you can tell superficially what is SVD, what is the meaning, right? Try to understand that from that particular lecture, last lecture of this, right? So is there any question about this week, second week? Okay, if there is no more question, then I will come to the short discussion on the week 3. What we are doing here, we are asking you to get habituated. Weekly, we are starting R. So you are supposed to get habituated with R and also learn the basic operation, arithmetic operation, logic operation, data reading, data writing. These are essential. See, there are many, if you pick up an R book, lots of things are there. I have tried to distill those things which are required to start. Step by step, we learn complicated thing in future also, right? So don't get lost in a book of R or something. See, every writer writes those books based upon certain target audience in mind. So when I have created this course, I have also kept in mind that the people who are using this course possibly have never used any programming language. For them, running R is very stable, right? Even installing R can be very difficult for some people, right? So we are learning step by step. So as I said, all these codes are available. Download them, play with them, and learn step by step what we are learning in these. Also, we will know we are teaching how to do the matrix and vectors, and how to manipulate them, how to calculate determinant, transpose of a matrix, inverse of a matrix using R. So try to do them paper and pen as well as in the R. And then data hacking. How to create data frame, how to create matrix from data frame, how to fetch a file from a location. All these are essential in R, right? So I try to teach them in this third week. Hope you will be able to go through them and learn this basic so that you can join me to learn much more complicated thing in the coming weeks. Okay, that's all from my side. If you have any more query, you can ask me here. Any question if you have, please ask either in the chat of the YouTube or here in the chat of Teams. So let me jot down a few things. One, check all videos and PPTs, right? The slides. Don't miss. Go through them repeatedly. Please go through them repeatedly. Second, attend live sessions. Every Saturday, right? Every Saturday, we have the live session. Please attend them. Note down the problem solved there. Try to solve them again. Again, okay, obviously, this is those live sessions are recorded and posted in YouTube. Go through them repeatedly. Okay? If you do these two things, lots of things will get clear. You'll easily go through the code. Third, if you have problem, you fail to understand something or query, post on forum. Right? Or send email to me. Right? Or by ask in live session. And the sixth most important thing, submit the assignment. Don't miss them. Don't miss a single assignment. Submit them. Even if you cannot answer all questions, try them. There's nothing wrong. And submit it. And I can guarantee if you do follow these instructions as I'm giving you, I've seen over the time, you can talk to other people. So if you have connections or your seniors or your friend who has done these courses earlier, believe me, if you follow these instructions, it's very easy to pass through this course and get a very good grade in the final exam. Any more questions? Yes, that's what we have, right? We are learning machine learning algorithm thing in the where it is. Let me check out where is my syllabus. Here it is. So week 4, you are doing data visualization because that is not part of any what we say machine learning. But then week 5, correlation regression. Although people do not realize regression models are actually machine learning, right? So you start with regression model, linear, non-linear. Then week 6, again, we are going into another machine learning thing, clustering, classification. We'll continue in week 7 with clustering and classification. And week 8, we will have dimension reduction, which is again a machine learning. Right? Sir, being a biochemistry student, how can I memorize these codes of R language? You do not memorize. It is a programming language, just like any other language you learn by using, right? You do not memorize English language. You have learned it by using repeatedly. The more you use, the more you get habituated, right? So this is true for your mother tongue also. So if you look into the language of R, thick books will be there. You cannot learn everything, all grammar and syntax, every function of R. Don't try that. Go through what we are teaching, use them repeatedly. That's why I say, download the code that I am using in the lecture, use them, manipulate them. Even if you don't understand, remove one bracket, remove one colon, see what is happening. Think in a different way, how can I write the same line, right? Same script. Believe me, if you practice it regularly, automatically it will become a natural language for you. It does not take much time. The only trick is practice. How to practice? Take a code, break it, play with it, right? So don't try to memorize, that will not help. Second big session, the rank of a matrix or practice I came across reduced row, which was kindly ignored. Reduced row echelon is a big topic. I will not go into that right now, right, Salma? It is a big topic. It will take some more time, and I have to give you a step by step, take an example and step by step you have to show them, right? So I don't think that may help you if I can do that. I do not have time today also, but in general that does not help the better audience of this course. That's why we have not discussed that. If you are good in math and linear algebra, if you have done linear algebra earlier, then I'll try you to try that. Otherwise, don't get lost into all these things. Only thing in terms of rank we have to remember is that, see, if the if the all the columns and rows are linearly independent, then the number of columns or row which is the maximum, minimum is the rank of the matrix, right? And whereas if they are not independent, then the rank is not full rank, right? The number of column and the number rank does not match, rank will be less. And if your rank is not full rank, then you cannot calculate determinant. You can calculate determinant, but it is zero, so you cannot do inversion and other thing, right? So if you have done, if you are comfortable with linear algebra, then we will ask you. There are standard textbook, online material also on row echelon. You can try that. But otherwise, if you have not, I will insist that you may avoid it now, and it doesn't really help you in this course. Fine. Okay. Now, upcoming, is there any probability and stats problem in R? Yes. In this third week only, we are dealing with some stats problem in R. How to do t-test and ANOVA, right? So check them, right? And then we are going to get any project related to our doing the note. We are not giving any project separately. That will overburden the students. What I insist that whatever are we are using in the lecture, please go through. I hope that helps. Okay, we have 1 or 2 minutes if you want to ask any more questions. Okay, so if there is no more question, final exam is always 100 marks, right? Marks does not matter. You can go to the landing page of the course where details of calculate how final marks is calculated is given, how much percentage comes from the assignment, how much comes from the final thing, all are given there. So in general, how much is the final doesn't really matter because things will be scaled anyway. A percentage will take. So you can go through the course page, there details of the exam pattern and everything is given. Right? Okay, then we'll close this session. Almost 1 hour is over. I hope I could briefly give you the essence of this course and the key point that you should keep in mind to go through this course and succeed in the final exam, right? I hope you will stick to this. Even if you feel some difficulty, share that difficulty to this course and persist in the course. Submit the assignment and attend register for the final exam, give the final examination. I hope you will enjoy this course and possibly I'll see you again in another live session. Till then, happy learning. Thank you.